Okay, this is the video for medical interventions for December 11th and December 14th. And it's going to go through what we're going to be doing in class and what you should be doing at home when you're not in class. So let's look in Canvas first. And we're almost to winter break, so keep hanging in there and we'll be getting through this. So we're going to finish up. We're going to do the next step in the Superbugs lab in class. So hopefully at this point in this workbook, you have described the difference between the strains. You have described mutation, inactivation, destruction, and efflux based on the video, and I'll clarify any questions you have about that in class. And that you've also made predictions about what's going to grow on each plate. Spoiler alert here, but if you want to go ahead and see what they should look like, these are what the results should look like. Okay, the plate with LB, you should see growth on both. The plate with ampicillin, the strain number 2 grows because it had the AMP resistance gene. You should have growth of E. coli 1 on the streptomycin plate and no growth on either because it had both strep and AMP on the whole thing. So neither one of them had resistance to that. So results look pretty good. So that's what you guys should have ended up with. And so we're going to record those results or what you actually see in class. And then we're going to do the next step of it. We won't be ready to do this final plates yet because we won't see those final plates just yet. We won't see those until Tuesday, Wednesday in class. And then you'll have a write-up to do for that. So the only other thing I want you to do at this point is to hold on just a second. Sorry. The only thing I want you to do now is to look at mechanisms of gene transfer among bacteria. And so here, for each one, label the picture with the term and a description of how it happens. So if you go back to the FDA video, that's in 1, 2, 3, all the way down, farther, the same video that we watched last time. And you watch the same video that's in here, and it describes antimicrobial resistance. Later in the video, the genetic transfer. So there's 3 methods: conjugation, transformation, and transduction. So those are the 3 terms. All right, so those 3 terms will go with one of these 3, each one with one of those with a description. All right, and then we'll talk more about this, so you won't be able to do this yet. So for the 1, 2, 3 assignment, what you need to be doing is basically these are final results. So these are predictions that we'll be doing in class, so you don't have to worry about that. But outside of class, really, all you need to be doing is make sure you have this box done and make sure you have this box done. Okay. So we're going to move on to 1, 2, 4 in class also, because this next step really in the Superbugs class is very simple. All you're going to do is take cells, resistant cells. Okay, we're going to make what's called a mixed plate. Okay, and so we're going to have one LB agar plate. So this is from our initial growth. We're going to take one colony of E. coli 1 that is resistant to streptomycin, and one colony of E. coli 2 that is resistant to ampicillin, and put them apart from each other on a mixed plate. Okay, and so from there, we are going to try to allow them to transfer resistance one to the other. And we're going to mix those strains together. So you're going to start by mixing one of the colonies towards the other, and then from there you streak it out. What we don't want to do is eventually, what we're trying to do is then we're going to take a sample of this on the next day from our mixed plate, a single colony from there, and streak them onto the different antibiotic agars and see if they can grow. And so that's what our plan is going to be for that. So we're making this mixed plate. This takes about 5 minutes to do in class, so it won't take very long. So that's the next step in that, but we have to wait then for them to grow. So we will then view the final results on Tuesday and Wednesday. Just a heads up, on Tuesday and Wednesday, you will be given an assignment for this 1.2.3 assessment. I've not posted it in Canvas yet, so you don't have access to it yet, but you will later in the week. And there's a lab analysis and conclusions that relate to the concepts in general, as well as like what you found out. So you're describing the evidence, what you think happened, how you know, and your reasoning that connects to evidence. So I'll explain that more next time, but just understand that's a big assignment. So don't need to do anything with that. So really, 1, 2, 3, you're explaining the mechanisms of transfer and the mechanisms of resistance. Might have some of that done already. So most of your time outside of class is going to be spent on 1, 2, 4. And we're going to be doing a lot of this in class as well. But if you happen to not be here at all, you can do this assignment at home. So the one thing I want you to do with 1, 2, 4 is we're looking at what happens if you don't take your antibiotics. So when antibiotics fail, okay? So if someone's prescribed an antibiotic, they're told to finish the entire prescription and to not stop once you start feeling better. All right? And they may tell you to take it for 7 days or 10 days or 8 days. And so we're going to use little colored disc, like bingo chip things in class. But you certainly don't need to have that because you could just, it's a visual, but you could do the counting yourself. And so you're going to do one trial. The one thing I'd like you to do on your own time at home, if you're on the gold day, or if you're not here first, or at all, is to do the trial for someone who does take their antibiotic every day. Okay, and so each person will be given these discs, blue, yellow, and red discs, things like little chips. All right, blue represents the least resistant bacteria, which means they're more easily killed. Pneumococcal bacteria, which causes pneumonia. Yellow discs are resistant, and red discs are extremely resistant. Okay, and it says toss a number cube, dice, right? To do that. So you can use dice at home or whatever if you're not going to be in class. If you're in class, we will do this simulation in class. So what I want you to do in the 1, 2, 4 activity, so if you go to 1.2.4, when antibiotics fail, is you're going to do the perfect trial, all right? And just kind of get the data and the graph filled out on that. So the initial counts is that of the 20 bacteria colonies, or let's say there's 20 bacterial cells, which is way lower than what there would be. There are 20 bacterial cells, 13 of them are blue, 6 are yellow, and 1 is red. So essentially, if you do the math, like 65% are not very resistant, 30% are resistant, and 5% are very resistant. So here's how this works. If you look at the directions, when you roll the dice in the real simulation, all right, we're doing the perfect trial first, so we can kind of compare that. If you get a 1, 3, 5, or 6, you took the antibiotic on time, so the bacteria are killed. All right, and you remove 5 discs, okay? But you start by removing blue discs first until they're all gone, and then move to yellow, and then red, and so on. When you get to the other part of the simulation, you get a 2 or 4. That means you forgot to take the antibiotic, and so you don't remove any. So any bacteria then that are still alive, you add 1 disc of that color, just 1 disc, not per bacteria, but 1 disc of that color for of bacteria, a particular type, are still alive, and that shows them reproducing. Okay, and then yes, you add 1 of that color. So for instance, in this case, they're still going to be reproducing, but their numbers are going down. So for instance, in this first one, if you take the medication, all right, then you're going to and you record that number. The key on this one, let me go back and double check that. Record blue, yellow, and red discs now in the population. So you're going to take 5 away because you took your medication, but you're going to start with the blue. And so your next generation would have 8. Then these are still 6 because we haven't killed them yet, and this is still 1. So now you're at 15. But, and here's the tricky part, is before they do reproduce. So then essentially you add 1 to each generation population that's still left. So essentially that's 9. And this would be 7. And this would be 2. All right? Because you add 1 for each population for each new trial. Then you roll the dice again. In this case, this is a perfect trial. So In this case, it's a perfect trial. And then you'll get your numbers until basically you kill all of the bacteria in this trial. And you're going to track those. And what you're going to do is you're going to graph the individual strains, not the total bacteria. So on the graph, x-axis, the independent variable, is the different type of, is the day, essentially. The toss number is the day. So like day 1, day 2, day 3, day 4, right? And then this is the number of bacteria remaining. You would have a blue line, a yellow line, and a red line to demonstrate those bacteria. So those resistant ones might be growing initially, and then they will eventually drop once there's very few of them left. And then they become sort of overwhelmed by the antibiotic, and they don't have it. There's not enough population for them to continue to grow. So if you would do the perfect trial, this trial outside of class, and if you're in class, we're going to do this random trial together. All right, so you have those results to compare to, and then you have a little bit of a write-up on that. Let's do it at the end of next week as well. So we'll be doing this part together in class. If you happen to be gone the whole time, you happen to be gone the whole week, or are unable to come to class for this, then what I want you to do is, if you have a dice at home, you can do this and follow the directions that are in the activity, where 1, 3, 5, or 6 mean you took your antibiotics, you do what we just did. But if you get a 2 or a 4, you forgot the antibiotic. So just to kind of simulate that, if I got a 2 or a 4, I forgot to take it, so I do nothing. So let's say that first one I got a 2 or 4, so I do nothing, and so now I grow them each by 1. 14, 7, 2. Now I have 23, okay? And then I go from there. So each time you don't take antibiotics, it's going to increase the number that you have. So do the best you can with that. I can answer questions on that more on Tuesday, Wednesday too. But this assignment is due Thursday as well. At the end of the day, 11 to 10 p.m. Thursday, along with the 1, 2, 3 assessments. So really, next week, Friday, Monday, we have our 1.2.3 lab, 1.2.3 assessment, 1.2.4 lab, and that's pretty much it. All right. So, and if you don't have a dice at home or something like that, you can go to an online timer. If you actually go to the distance learning activity version of this, it has a link to an online stopwatch, and as a virtual die, which you can just Google that as well. And you can also click that and get a number. And don't cheat. Don't cheat. Do it. Be fair. If you don't take your antibiotics, you have to deal with consequences. So, all right, that's it. And let me know if you have any questions. Thanks.